Welcome, everyone. I'm excited to kick off today's presentation on wood's softwood anatomy. This will be our first slide, setting the stage for what's to come. Over the next few slides, we'll explore the fascinating structure of softwoods, how they differ from other types of wood, and why understanding their anatomy is so important. So, let's get started and dive into the world of softwood anatomy. Let's talk about longitudinal tracheids. These cells are always present in the wood tissue, and they make up a significant portion, about 90 to 95% of the cell volume. They are long and thin, typically ranging from 1.5 to 5 millimeters in length, with a diameter between 20 and 70 micrometers. Their main functions are providing mechanical support and conducting water and nutrients through the plant. You'll notice that between tracheids, there are bordered pits, which facilitate water movement while preventing air bubbles from spreading. Between tracheids and parenchyma cells, there are semi-bordered pits, allowing selective communication between different cell types. Additionally, these tracheids show interesting features like transitions between early wood and late wood, which influence the wood's properties. Some tracheids also exhibit spiral thickening in their cell walls, adding extra strength and flexibility. All of these structural features work together to support the plant's survival and efficient transport, tying into the broader theme of how plant tissues are specialized for function and resilience. Today, let's take a closer look at the morphology of longitudinal tracheids. As you can see in the slide, we have two main types to consider: latewood and earlywood. These terms describe different parts of the annual growth ring in wood. Earlywood is the first part to form at the beginning of the growing season. It typically features larger, thinner-walled cells that are designed to maximize water conduction. Latewood, on the other hand, forms later in the season. Its cells are usually smaller and thicker-walled, providing more structural support. Understanding these differences is crucial because they reflect how the tree adapts to seasonal changes. Recognizing the distinct characteristics of earlywood and latewood helps us grasp how trees optimize both transport and strength throughout the year. This morphological variation is a key aspect of how wood functions in nature and in various practical applications. Let's take a closer look at the transition within a softwood growth ring, which is a key concept in understanding wood anatomy. Here, we see two examples: the eastern white pine and the southern yellow pine. These species exhibit different patterns of transition within their growth rings. In the case of the eastern white pine, the transition from earlywood to latewood tends to be more gradual. This means the change in cell structure and density happens slowly, resulting in a smoother transition. Conversely, in southern yellow pine, the transition is often more abrupt, with a sharper change between the earlywood and latewood. The diagram also illustrates this concept with the terms U for earlywood and LW for latewood. The transition can be either abrupt or gradual, and understanding these differences is crucial because they influence the wood's properties, like strength and appearance. Recognizing these patterns helps us identify species and assess their suitability for different applications. Overall, understanding how these transitions occur within growth rings is fundamental to our broader study of wood characteristics and their impact on use and performance. Let's take a look at Slide 5, which focuses on the ULW transition, often described as abrupt. This transition refers to the boundary between earlywood and latewood within a growth ring. As you can see, the transition can sometimes be quite sudden, which indicates a rapid change in growth conditions. Now, earlywood is the part of the growth ring that forms at the beginning of the growing season. It's typically lighter in color and less dense because the tree is actively growing and producing cells that are larger and more porous. Latewood, on the other hand, forms later in the season. It tends to be darker, denser, and more compact as the growth slows down and the cells become smaller and thicker. Hoadley's work helps us understand these transitions better, especially when they're abrupt. Such abrupt changes can tell us a lot about environmental shifts or stressors experienced by the tree during that period. Overall, recognizing these features is crucial for dendrochronology, as they help us interpret past climate conditions and growth patterns. So, Understanding the nature of this transition, whether abrupt or gradual, gives us valuable insights into the tree's history and environment. Let's take a closer look at the ULW transition, which occurs gradually within the tree ring. This transition reflects how the tree shifts from earlywood to latewood during the growing season. Earlywood, as the name suggests, forms at the beginning of the growing season and tends to be lighter and less dense. As the season progresses, the tree starts producing latewood, which is darker, denser, and forms later in the season. Hoadley's work provides important insights into how these transitions happen smoothly rather than abruptly, which is crucial for understanding the tree's growth patterns and environmental responses. Recognizing this gradual change helps us interpret annual growth rings more accurately, especially when analyzing climate conditions or wood properties. So, in essence, this slide emphasizes that the transition from earlywood to latewood is a gradual process, and understanding this helps us connect the physical characteristics of the wood to the tree's seasonal growth cycle. This concept ties into our broader theme of how tree rings serve as valuable records of environmental history. Now, let's take a closer look at the ULW transition, which is an intermediate stage in the wood formation process. During this phase, we see the formation of latewood, which is characterized by denser, smaller cells that contribute to the strength of the wood. On the other hand, earlywood is formed at the beginning of the growing season and has larger, less dense cells that allow for efficient water transport. Understanding the distinction and transition between earlywood and latewood is crucial because it reflects how the tree adapts to seasonal changes and how the wood's properties develop over time. This transition not only influences the overall structure and quality of the wood, but also ties into the broader theme of how trees grow and respond to their environment throughout the year. Today, I want to focus on how softwood cells are organized within the wood structure, specifically looking at their arrangement in radial files. When we talk about radial files, we're referring to the vertical columns of cells that run from the center of the tree outward. These files are aligned along the radius, which is the line from the core of the tree to its outer edge. This arrangement is key because it influences how the wood grows and how it responds to stresses. Now, alongside radial files, we also have tangential orientation, which runs perpendicular to the radius, following the curvature of the tree's growth rings. Understanding the difference between these two orientations, radial and tangential, is important because it affects the properties of the wood, like strength, shrinkage, and how it reacts to environmental factors. In essence, the organization of softwood cells in these radial files contributes significantly to the structural integrity of the wood. By studying these arrangements, we gain insights into how wood develops and how we can better utilize it for various applications. This connection between cell organization and wood properties really highlights the importance of microscopic structure in the overall performance of timber. So, when we look at hardwood cells, one interesting thing to note is that their alignments are actually less linear compared to other types of cells. Instead of being perfectly straight or uniformly arranged, these cells tend to have more irregular patterns. Now, if we break down their orientation, we see two main directions, radial and tangential. Radial refers to the direction that runs from the center of the tree outward, essentially along the radius. Tangential, on the other hand, runs tangent to the growth rings, so it's more around the circumference of the tree. These variations in cell alignment are really important because they influence the wood's properties, like strength and how it responds to stress. Understanding these differences helps us appreciate the complexity of hardwood structure and how it impacts everything from growth to practical uses. Now, let's take a closer look at the softwood bordered pit. This is a crucial structure in softwood anatomy. Essentially, it's a specialized feature that allows water to move efficiently between cells while maintaining structural integrity. The bordered pit is characterized by a thickened, framed border around the pit membrane, which helps regulate water flow and prevents air bubbles from spreading. This design is particularly important in softwoods, where efficient water transport is vital for the tree's health. Understanding these pits helps us appreciate how softwoods adapt to their environment and maintain their function. So, in summary, the softwood bordered pit is a key adaptation that supports water movement and overall tree vitality. Now, let's take a closer look at some of the key features found in plant cell walls. First up is the torus. This is a thickened, often circular region within the bordered pits, which are specialized structures in the cell wall. The bordered pits themselves are crucial because they allow for the movement of water and nutrients between cells, acting like tiny gateways. Margo, which is the region surrounding these pits, plays a vital role in controlling these exchanges. Understanding how these components work together helps us appreciate the complexity and efficiency of plant vascular systems. These structures are essential for maintaining plant health and facilitating the transport of fluids, which ties back to the overall theme of plant support and nutrient distribution. Now, let's talk about the longitudinal parenchyma. It's not always present in all species, and when it is, it's never found in large quantities. In fact, it makes up only about 1 to 2% of the cell volume. These cells are quite small and thin, typically measuring between 50 and 150 micrometers in length and 20 to 30 micrometers in diameter. What's interesting is that these cells can remain alive in the sapwood when they're functioning, which is important for their role. Their primary function is storage, and they are connected by simple pits between them. These pits are usually dispersed throughout, but sometimes they form boundaries. Certain trees always have longitudinal parenchyma, such as taxodium, sequoia, cupressus, and thuja. Others, like larix, pseudotsuga, tsuga, and abise, sometimes have it, but it's absent in species like pinus and picea. Understanding the presence and characteristics of longitudinal parenchyma helps us appreciate the diversity in wood anatomy and its implications for wood function and durability. Let's take a closer look at the softwood longitudinal parenchyma. These are the specialized cells found within softwoods that run parallel to the tree's axis, essentially along the length of the wood. They play a crucial role in the tree's physiology, particularly in storage and transport of nutrients and other substances. Unlike vessels, which are more prominent in hardwoods, these parenchyma cells are more characteristic of softwoods. They help in the storage of carbohydrates and support the overall health of the tree. Understanding their structure and function gives us insight into how softwoods grow and maintain their vitality. This is important because it ties into the overall theme of wood anatomy and how different cell types contribute to the properties of various wood species. Today, we're focusing on the concept of the longitudinal parenchyma, as discussed by Parenchyma and Dizio. The longitudinal parenchyma refers to the specialized tissue that runs along the length of certain structures in the body, providing both support and functional capacity. It's a key component in understanding how various organs maintain their shape and perform their specific roles. What's particularly interesting is how this tissue type aligns with the overall theme of structural specialization within the body. It highlights the importance of organized, directional tissue arrangements in ensuring proper function. As we move forward, keep in mind how these longitudinal systems integrate with other tissue types to create a cohesive and efficient biological framework. Now, let's talk about ray cells. In most species, they are uniseriate, meaning they consist of a single cell layer. However, in species like sequoia and cupressus, you can find biseriate rays, which are made up of 2 cell rows. The primary functions of these rays are transport and storage, playing a vital role in moving nutrients and storing reserves within the wood. Focusing on ray parenchyma, these cells can vary quite a bit in height, typically ranging from 2 to 40 cells tall. They have simple pits between themselves, which facilitate movement of substances from one ray cell to another. Also, there are semi-bordered pits with longitudinal tracheids, known as cross-field pits, connecting rays to the surrounding tracheids. Inside these ray parenchyma cells, you'll find cell contents, obviously, since these are living cells. Now, moving on to ray tracheids, these are usually arranged in 1 to 3 rows at the margins of the rays, but sometimes they can be present throughout the entire ray, especially in some species where they make up the whole ray structure. Ray tracheids also have bordered pits between them, which are crucial for water movement. These pits can have different wall structures, Some are smooth-walled, while others are dentate, meaning they have a toothed or irregular border, which can influence how water passes through. Finally, I want to highlight that in many species like Pinus, Pseudotsuga, Picea, Larix, Tsuga, Thuja, and Chamaecyparis, these ray tracheids are quite prominent. They help in maintaining the overall function of the ray system. So, understanding these components, the ray parenchyma, ray tracheids, and their pit structures, gives us a comprehensive view of how rays contribute to wood physiology. Now, let's turn our attention to softwood rays, an important feature highlighted in this slide. As Haygreen and Bowyer explain, these rays are essentially radial structures within the wood that play a crucial role in the tree's physiology. They are responsible for storing and transporting nutrients and water horizontally across the growth rings. Cathay also emphasizes that the size, distribution, and appearance of these rays can vary significantly among different species, which can influence the wood's properties and its suitability for various applications. Understanding the characteristics of softwood rays helps us appreciate the complexity of wood anatomy and how these microscopic features impact the overall strength, durability, and workability of wood. So, in the context of our broader discussion, recognizing these subtle but vital components allows us to better assess wood quality and make informed decisions in forestry and wood utilization. Let's take a closer look at the softwood ray structure. Rays are these thin, ribbon-like cells that run perpendicular to the growth rings, and they play a crucial role in the transport of nutrients and storage within the tree. In softwoods, the ray structure tends to be relatively simple and less prominent compared to hardwoods. This simplicity is important because it affects how we identify and differentiate softwoods from hardwoods under the microscope. Understanding the ray structure helps us grasp the overall organization of softwood anatomy, which is essential for applications like wood identification, processing, and even understanding the wood's mechanical properties. So, when we look at softwood rays, we're essentially seeing a key feature that links to the tree's functionality and its practical uses. This connection underscores the importance of anatomy in forestry and wood science, tying back to the overall theme of how structure influences properties and applications. Let's take a moment to explore the insights from Hoadley and Cathay. These researchers have shed light on some key aspects that are crucial to our understanding. Hoadley's work emphasizes the importance of contextual learning and how people engage with content in real-world settings. It's about making sure that learning is meaningful and applicable. On the other hand, Cathay focuses on the social dimensions, how relationships and community involvement influence motivation and persistence. Both perspectives highlight that effective learning isn't just about individual effort. It's also about the environment and social context. Connecting these ideas to our overall theme, we see that fostering engaging, supportive environments can significantly enhance learning outcomes. This understanding helps us design better strategies that align with how people actually learn and stay motivated. So, on this slide, I want to highlight some of the key points that tie into the overall theme of our presentation. First, let's look at the importance of understanding the core concepts thoroughly. This foundation is crucial because it allows us to make informed decisions and apply these ideas effectively in real-world situations. Next, I want to emphasize the significance of engaging with the material in a conversational way. When we approach these topics as if we're having a dialogue, it helps solidify our understanding and makes the learning process more dynamic and enjoyable. Now, a critical concept here is connecting individual points to the bigger picture. It's not just about isolated facts. It's about seeing how everything fits together and supports our overall objectives. Finally, I want to remind you that this approach, being thorough, conversational, and connected, is what makes our strategy impactful and sustainable. It's about integrating these ideas seamlessly into our daily work, ensuring they truly resonate and lead to meaningful results. Now, let's take a closer look at what Jossa mentioned here. He talks about pitting on the cell walls of longitudinal tracheids. What does that mean exactly? Well, pitting refers to specialized areas in the cell wall that allow for water movement between cells. In the case of these tracheids, which are elongated cells responsible for water conduction in plants. These pits are crucial. They enable water to pass efficiently from one tracheid to another, maintaining the flow throughout the plant. The term longitudinal highlights that these tracheids run lengthwise, aligning with the direction of water movement. So, understanding these pits is key because they play a vital role in the plant's ability to transport water effectively, which ties back to the overall theme of how plant vascular systems function to support growth and survival. So, let's take a moment to really understand these key points. First, it's important to recognize that effective communication is at the heart of everything we do. When we communicate clearly, we can avoid misunderstandings and foster stronger relationships. Second, I want to highlight that active listening plays a crucial role here. It's not just about speaking, but truly engaging with what others are saying, which helps us respond more thoughtfully and build trust. Third, connecting these ideas to our overall theme, which is about enhancing collaboration and productivity, shows that good communication isn't just a nice-to-have, it's a strategic asset. When we master these skills, we set the stage for better teamwork, innovation, and success. So, keep these concepts in mind as we move forward. They are the foundation for everything else we're aiming to achieve together. Let's take a closer look at resin canals. These are intercellular spaces, meaning they are not made up of cells themselves, but rather gaps between cells. They can be oriented both longitudinally and transversely within the wood. The primary function of resin canals is protection, helping the tree defend itself, especially against pests and pathogens. You'll find these canals in species like Pinus, Picea, Pseudotsuga, and Larix. Sometimes, resin is also called pitch, and you'll see that term used interchangeably. Moving on to fusiform rays, these are composed of epithelial parenchyma, ray parenchyma, and ray tracheids. They play a vital role in the radial transport of nutrients and storage within the wood. Now, traumatic resin canals are quite interesting. They form in response to injury or stimulus, essentially as a defense mechanism. These are present in regular resinous species, as well as in tsuga, abais, and sequoia. Unlike the regular resin canals, traumatic ones are only formed longitudinally. You can think of them as being created vertically within the wood. They also contain epithelial parenchyma, which lines the canal, whether it's a vertical resin canal or a horizontal one. Understanding these structures helps us see how trees protect themselves and manage their internal chemistry, key concepts in wood anatomy and tree physiology. Resin canals are these fascinating tubular passages found within the wood, and what's interesting is that they are actually intercellular spaces. They're surrounded by specialized parenchyma cells, which we call epithelial cells. These cells play a crucial role because they exude resin or pitch into these canals. Now, why is this important? Well, these canals serve as a protective mechanism for the tree. When the tree gets wounded, whether from mechanical damage or boring insects, the resin or pitch is exuded into the canals to help seal off the wound and prevent further harm. If we look at specific examples like Douglas fir or pine, we can see these resin canals running longitudinally through the wood. The epithelial parenchyma cells line these canals, facilitating the flow of resin. This system is a vital part of the tree's defense strategy, helping it to survive and recover from injuries. So, in the bigger picture of wood anatomy, resin canals are a key feature that highlight how trees have evolved mechanisms to protect themselves from external threats. They're integral to understanding both the structure and the resilience of wood. Let's take a closer look at resin canals now. These tiny structures are crucial because they produce and store resin, which plays a vital role in the tree's defense mechanism. If you look at the slide, you'll see that different species have distinct types of resin canals. For example, in white pine, the resin canals are typically located in the fascicles along the needles. Meanwhile, lodgepole pine also has resin canals, but their arrangement and density can vary. Moving on to spruce, you'll notice they tend to have resin canals distributed throughout their wood, which helps in healing wounds and deterring pests. Lastly, Douglas fir features resin canals that are mostly found in the rays and some in the tracheids. Understanding these differences is important because it helps us identify species and also assess their resilience against pests and diseases. All these details tie back to our overall theme of understanding wood anatomy and how it influences the properties and uses of different tree species. Now, let's take a closer look at resin canals. These are quite fascinating because they come in different sizes. On this slide, you'll see that we have large diameter canals, which are often more prominent and easier to identify. Then, we also have small diameter canals, which can be a bit more subtle but are equally important. The terms "cate" here refer to the different sides or aspects of the tooth where these canals are located. Understanding the variation in canal size and their orientation is crucial for effective treatment, as it influences how we approach cleaning, shaping, and filling during procedures. Recognizing these differences helps us ensure a more thorough and successful outcome. So, always pay close attention to these details. They're key to understanding the complex anatomy of the resin system. Today, I want to focus on the rays, specifically fusiform rays. These are a particular type of ray characterized by their spindle-shaped structure. When we look at fusiform rays, we see that they have a distinctive shape that is wider in the middle and tapers towards the ends. An important feature to note is that these rays often include a horizontal resin canal. This canal plays a crucial role in the plant's defense mechanism, helping to transport resin that can deter herbivores or heal wounds. Recognizing fusiform rays and their resin canals is key to understanding plant anatomy and how these structures contribute to the plant's overall resilience. This detail ties back to our broader discussion on plant adaptations and structural features that support survival in various environments.